You've seen the headlines, right? Scientists extend lifespan of mice. For decades, it sounded like something straight out of science fiction, a dream just around the corner. But you have to ask, where is it for us? Well, in this explainer, we're going to dig into why that dream has been stuck in the lab for so long and how a revolutionary breakthrough might finally be about to change, well, everything.
I mean, this is the billion-dollar question, isn't it? For nearly 100 years, we've proven we can slow down aging in animals. So why not in humans? What's the holdup? Why has all that progress for our own species just hit a brick wall for so long?
Okay, so first things first, let's be crystal clear. Being able to manipulate aging in a lab isn't some fantasy. It's a well-established, decades-old reality. And the success scientists have had with lab animals, it has been nothing short of astonishing. And look, this is not some new thing. This whole journey started way back in 1934 with a pretty simple discovery. Just by restricting calories in rats, they lived up to 40% longer. Just think about that for a second. That was the first hard proof that aging wasn't some inevitable fixed countdown. It was malleable. And that one discovery kicked off a whole century of research, leading to huge government programs and blockbuster drugs like rapamycin, all proving the same thing over and over again in the lab. We can intervene in the aging process. So with all this incredible success in the lab, what's been the big roadblock for humans? You'd think it would be some super complex biological mystery, but it turns out the answer is a surprisingly practical problem. And here it is. This is the single biggest hurdle that's held everything back: the measurement problem. I mean, think about it. How do you prove an anti-aging drug actually works on a creature that can live for 80, 90, maybe even 100 years? You can't just run a clinical trial for decades waiting for people to, age and die. It's just wildly impractical, impossibly expensive, and for generations it brought human research to a complete standstill. To get around this massive problem, we have to understand a really critical difference. There are two types of age. First, you've got your chronological age. That's easy, right? It's just the number of candles on your birthday cake. But then, there's your biological age. And this is the number that truly matters. It's the real wear and tear on your body systems. A great analogy is a vintage car. Its model year, that's its chronological age. But the actual condition of its engine, its transmission, its frame, that's its biological age. And that's the number scientists desperately needed to be able to measure. And this right here is where the entire story completely shifts, because in just the last few years, a monumental breakthrough has finally given researchers the tool they've been missing for a century: a reliable way to measure biological age, not in decades, but right now. They're called aging clocks, and they are an absolute game changer. Seriously, forget what you know about simple blood tests. These are incredibly sophisticated tools, using powerful algorithms to scan huge amounts of your personal biological data to calculate one single crucial number: your true biological age. And just like that, the biggest roadblock in the history of anti-aging research, it pretty much vanished. And the amount of data these clocks are crunching is just staggering. They aren't just looking at one thing, they're looking at everything. They're powered by machine learning, and they analyze the chemical markers on your DNA, the proteins floating in your blood, the activity of your genes, even the specific types of bacteria living in your gut. It is like getting a full-body diagnostic at the molecular level, all focused on answering one question: how well are you really aging? So the big question is, are they actually accurate? And the answer is, yeah, remarkably so. The average error rate for these clocks is only about 2 to 8 years. Now that might sound like a big range, but over an 80-plus year lifespan, that's a tiny fraction, just 5 to 10%. And that level of precision is more than enough to use in a clinical trial. All of a sudden, you can see if an intervention is working in a matter of months, not an entire lifetime. But here's why this is so unbelievably important. These clocks don't just tell you a number. They are powerfully linked to your mortality risk. If your aging clock says you're biologically older than your calendar age, your risk of age-related disease and death is measurably higher. That makes them the ultimate tool. If you can develop something that turns back the clock, you are making a real, meaningful impact on human health. Okay, so let's just recap for a second. The huge historical problem of measurement, it's solved. The revolutionary new tools, they're in our hands. So what's next? Well, this brings us to a bold call to action, a grand challenge for the entire scientific community.
The author of the paper that inspired this whole explainer, Morton Scheibye-Knutsen, he puts it very bluntly. He says, I propose a grand challenge to the field. Translate interventions from mice to trials in humans.
And that challenge is no longer some pipe dream, thanks to the power of these clocks. The math here is just astounding. Because the clocks are so precise, you don't need thousands of people for a trial anymore. In fact, a simple calculation shows that to see a statistically significant effect, like reversing someone's biological age from 75 down to 70, you might only need 16 volunteers. Just 16. That completely changes the entire game. Suddenly, human trials can be small, fast, and dramatically cheaper. So this all builds into a clear, actionable plan. Basically a human interventions testing program, modeled on those successful mouse programs. And the strategy is elegant in its simplicity. Step 1, you take a small group, maybe 50 people, and treat them with a promising compound for just 6 months. Step 2, you evaluate their biological age using these new clocks. Did you see a positive signal? Great. Then step 3, you just add another 6 months to the treatment and you reevaluate. It's a fast, iterative, and incredibly smart way to finally figure out what actually works in people. So where does all this leave us? Well, we are standing at a truly pivotal moment. With these new tools in hand and a clear roadmap ahead, we are walking into an entirely new era in the science of human aging. Now, of course, this doesn't mean we just stop doing research on animals. Not at all. That work is still absolutely critical for discovering new interventions, for initial safety testing, and for understanding the deep, fundamental biology of why we age in the first place. But really, the biggest takeaway from the last century of research is this profound idea. Aging is not some fixed destiny that's written in stone. It's a biological process. And because it's a process, it can be targeted, it can be influenced, and it can be changed. And for the very first time, there is a tangible, scientifically backed glimmer of hope. Which brings us to one last, really powerful question. For almost 100 years, we've known this was possible in a lab. Now, we finally have the tools to try it in humans. We've moved beyond if and straight into when and how. So are we standing on the brink of finally escaping the slow decay? Are we about to fundamentally change what it means to grow old?